All right, here we go. So my name is Angela Collins. I am with the University of Florida and Florida Sea Grant. I'm based at the Tropical Aquaculture Lab in Ruskin, which is in the Tampa Bay area on the west coast of Florida. I'm a marine fisheries and shellfish extension specialist, so I work closely with industry. And I'll be facilitating today's session, which is just basically an informational session about a new project that has started here on the West Coast. And I just want everyone to know that I am a resource for anyone on this topic or anything else shellfish related. So I'm here to help. Feel free to reach out anytime about this or anything else. Our research leads on this project are from the Virginia Institute of Marine Science, Kim Reese and Jan McDowell, who are both here and on camera in the same office. We've got them in the same space here. They are both professors at the Virginia Institute of Marine Science. Jan has been faculty there since 2005. Her research interests are focused on molecular biology and population genetics of marine organisms, including marine fishes, bivalve fishes, or bivalve shellfish, and even the pathogens that impact some of these species. Kim has also been working on genetics and specifically a lot of bivalve shellfish genetics for over 30 years. Her lab's work focuses specifically on molecular genetic analyses of a lot of shellfish species, including phylogenetics, population genetics, and genomics. And I'm going to put links to both of their lab pages into the chat here in a moment so that you can see some of the publications that they have produced in their decades of experience on this topic. Stephen is the director of the Gulf Shellfish Institute. He's based in the Tampa Bay region here on the west coast of Florida. For those of you who aren't familiar with the Gulf Shellfish Institute, it is a nonprofit organization focused on research to do environmental and commercial good, specifically focused on shellfish species in the Gulf. And honestly, it's the Gulf Shellfish Institute, but I think their work is applicable on both the Atlantic and the Gulf coasts. The GSI is leading the field portion of this project and working directly with industry partners to collect southern hard clams and integrate the objectives of the work with our industry here in Florida, including our shellfish hatcheries. The industry partner on this project is Kurt Hemmel, who's the owner and operator of Bay Shellfish Company. Bay Shellfish Company has been producing bivalve seed for both commercial and research purposes for over 25 years here, and they have an active invested interest in promoting aquaculture in Florida and throughout the U.S. Bay Shellfish Company is located again here in the Tampa Bay area, but they do provide seed to industry members across the state and even in other parts of the Southeast. So this proposal that we're going to be talking about today is funded through the National Sea Grant Aquaculture Initiative. It was submitted to an active RFP that came out in 2024. The funding was awarded early this year, and the project is scheduled to be completed around the end of 2027. So again, we're going to have lots of time for questions at the end. So in the meantime, if you have any, go ahead and stick them in the chat. But before we get started, I'd like to get a little bit of information from the folks that are on the call. So I'm going to put a little poll that's going to come onto your screen here in just a second. And it's just 3 questions. It'll take 60 seconds. So if you can just respond to the poll, it'll help us kind of get an idea of who's here. Are you seeing it? You got it now? Okay. So they're pretty quick questions. I'm going to count to 15 in my head and then I'll end the poll. Okay, great. Thanks for doing that, everybody. All right. So without further ado, I'm going to go ahead and let Kim and Jan take over here. And they are going to give you a little bit of an overview about the project as it's just now beginning.
Okay. Good afternoon, everyone. I'm going to talk to you basically more about the science behind this, and then Stephen's going to talk a little bit more about some of the collections and working with you to get your involvement and your feedback on some of this, and talk to you about how some of the industry may become involved with helping us with this project. So I'm going to talk about the development and the testing of what we're hoping will become an affordable and efficient, what we're going to call a marker panel or a genetic panel to advance the southern hard clam or Mercenaria campechiensis aquaculture in Florida. So the southern hard clam is distributed, as you know, along the Atlantic coast from North Carolina, but primarily in Florida and into the Gulf of Mexico, and it's considered the native Mercenaria species along the Florida's Gulf Coast. So we know that there's several multi-million-dollar state restoration projects that are funded in response to the public concern over the declining water quality in Florida. And so this presents a novel economic opportunity for hatcheries and growers in Florida because there's this interest in the native clam species. But the problem is that the northern hard clam, Mercenaria, or the quahog, was introduced to the Gulf Coast for aquaculture in the mid to late 20th century. And you know that Mercenaria and campechiensis readily hybridize. And there's no rapid method right now to distinguish the two species or to accurately determine the levels of hybridization, or what we call those F1 or F2 generations after they've hybridized, or the progeny, or the offspring, or the second generation offspring. So it results in this verified lack of broodstock. So what we have here on the right, and hopefully you can see my pointer, I hope, is up at the top. These are hatchery seed in this dark blue or purple color that are the northern hard clam broodstock. And then here in this teal color, these are wild clams from Tampa Bay, Florida. And you can see there the teals. So these are the campies and these are the quahogs. Okay, so you can see that those are probably the pure species of each type. And then you see here that there are some that have a little bit of the northern type and primarily the southern. Okay, and this one looks like it's a hybrid of about 50% of each. And then here's a wild clam from Alabama that's a campy. Okay, and here we have hatchery seed that was thought to be M. campi, like broodstock. Okay, hatchery seed from the broodstock that was thought to be M. campi, but it looks like it was really hybrid. Okay, and here we have hatchery seed from wild M. campis, and that looks like it really was campies. Right, so we see the problem here, and this was done using a large number of markers using sequencing. So this is not a rapid or efficient method of genotyping, but we're interested in developing a rapid and efficient method of genotyping, okay, that can be affordable and much more straightforward for the use by industry. Right now, Hatcheries rely on the external shell morphology to identify the southern hard clam broodstock. But the external traits, as we could see by the previous slide, are rather poor predictors of the underlying genotypes, or identifying which species is which. So without that confidence in identifying, the aquaculture industry cannot assess the commercial value, the true commercial value anyway, of the southern hard clam, of the campies. They can't determine if they're having northern hard clams or southern hard clams because of this mixture. They don't know if they have the hybrids, as we could see in the case where they thought they had M. campi broodstock and they spawned those and they ended up with actual hybrids as the offspring. Or they have back crosses are being spawned, or what's being released into the environment. So you have the regulatory agencies that are interested in doing restoration, but they may not be actually restoring with the native campies, okay? And then you have hatcheries that are interested in using campies and distinguishing those, from the northern hard clam, the quahogs. And these are you're having this confusion between the two species. So we need to meet this growing call for the native hard clam restoration in the Gulf states, as well as, probably a growing call, and you will know better than those of us from Virginia, but there's probably a growing demand for the native species, maybe even among your public. Maybe there's even a growing demand among the public for consumption. So the project objective here is to enable the growth of the southern hard clam in the aquaculture industry by developing this fast and affordable method to verify the genetic integrity of broodstock, so those broodstock can be used for spawning. So what are we going to do? We're going to collect samples from across the reported native Florida range, okay? And then obtaining those southern hard clam samples, maybe from Georgia and South Carolina if possible. Now this is very difficult because, of course, we're going to have to verify, first of all, using other methods and then sending them off for verification that they are actually campies and not quahogs. We'll have to isolate the DNA, genotype, and then we want to assess the population structure to establish a genetic baseline for actually the marker development to determine which ones we're going to use in our reduced marker panel to be able to distinguish more clearly. So this is where we get a little bit into the weeds about which genetic markers we're going to be using. So the markers that we're going to be using are called single nucleotide polymorphism, and that's a bit of a mouthful. So we just call them SNPs. Again, this is an acronym, but these are single base pair differences here in the DNA among individuals in their genomes or their DNA, okay? So if you look here, all these different clams, and you can talk about these different clam lines, they have this differences. Some of them have the red A, and some of them have here, as we've distinguished it, the blue C. So they're different at this particular site. And what we would be looking for is many of these differences to use as what we're going to call SNP markers, or differences that can be used to address the relationships or differences among individuals or clam lines or populations. And then ideally, we want to be able to use those to look at the species differences, just like we did before with a much larger set of markers. But we want to be able to identify key markers that we can use that are the best markers to use to be able to get to the point that we did here, remember with our diagram here, where we were able to say, okay, these are the cohogs, these are the campies, and these are the hybrids. And we want to have a good panel that's really efficient, very affordable to be able to distinguish those two species. So we're going to combine the data that we already have, and we have a good set of data already for the northern hard clams, and identify a set of SNP markers to discriminate those northern and southern hard clams and identify the hybrids in a very rapid, affordable, and be very confident in our diagnostic testing using a commercially available, and make it commercially available to the aquaculture industry. So you can ask us lots of questions about this later if that didn't make sense, and we can go over it a little bit more. So we're going to use standard protocols and a portal for the submission of tissue samples by hatcheries interested in broodstock verification through the Gulf Shellfish Institute website. And we're going to use a third party for DNA isolation. So they'll take the tissues and do the actual marker genotyping service out in, probably the service we'll use is a company out in California at a set cost at hopefully estimating only $12 or $17 per sample. That's the range they've given us, depending on the number of markers or SNPs that we need for accurate discrimination between the cohogs and the campies, and then produce a guide to interpret the genetic results that will be available on the GSI website and through Florida Sea Grant. And GSI and Bay Shellfish Company they're going to work with industry and the regulatory agencies to improve the sourcing and genetic verification of broodstock throughout the state. And so the participating hatcheries, which is part of what Stephen's going to talk to you about in a minute, will be offered genetically verified southern hard clam broodstock. And this is part of why we're going to be looking at different populations, different wild clams from different locations throughout the state from the closest genetic baseline sampling locations. So we're going to be getting wild clam samples also in order to know where wild broodstock can be sourced from. And GSI is going to work with hatcheries who have southern hard clam broodstock to begin the diagnostic testing and help source new individuals if a lot of the northern hard clam ancestry is found in those broodstock samples, which is quite possible based on what we know so far. So this is the timeline. We're right now in year 1. So in the process of collecting the samples, and we're going to be isolating the DNA and screening them using some other techniques that we have and sending them off for sequencing and identifying those SNP markers. And right now we're meeting with industry and determining interest in participation. And next year we plan to be finishing the screening and sequencing and identifying those markers for the panel and reporting back to you with our results. And then we hope to source and verify some selected broodstock in the 3rd year and communicate the materials and disseminate the results to industry and to the regulatory agencies. Jan, did you want to add anything?
No, I think that's great. I think we're off to Stephen.
I think that was a fantastic overview. And remember, we're going to have lots of time to pick their brain at the end. And I think there will probably be quite a few questions
we have a common brain
for both of you.
Brains. Thank you.
And thank you. Thank you so much for that, Kim and Jan. And then Stephen, do you want to go ahead and start? VIMS, there you go. If you can stop sharing.
Stop sharing. Yep.
Perfect.
Okay, can you hear me okay? Great.
Perfect.
Yeah, thanks for having me, everyone. I'm going to be talking a little bit more in depth about the collection process mainly because that's our job at the Gulf Shellfish Institute is to be the fins in the water and finding the specimens across the west coast of Florida. So I'm really excited about this project. Our team's really excited about the project because we get to travel all around the state and look for hard clams, which I don't think is a very bad gig at all. Um, just briefly, for those who might be listening in later, or who aren't familiar with the Gulf Shellfish Institute, as Angela already said, we're a nonprofit organization. We were founded in 2015, and our mission is to be provide research, scientific research that improves the shellfish aquaculture industry, but also the what we are calling now kind of restoration aquaculture. So utilizing the aquaculture process and merging the industry with all the environmental benefits that that shellfish can provide. We're composed of 10 board of directors from a very diverse set of expertise. The majority of our board of directors are active shellfish industry members, so we're very fortunate from that. But we also have some wonderful scientists and technical advisors as well. We've got 6 full-time staff, and we might be bringing on another for this project actually. But until then, the individuals in the green boxes are kind of the main people on the project. So myself, and then Sarah Hutchins is really going to be the project leader, and then Sarah Davis will be assisting with a lot of the in-water work. So for, on some of the other slides here, myself, obviously I'll put my contact information in the chat, but Sarah Hutchins is also an excellent person to talk to any questions about project moving forward. Okay, so Jan and Kim already did a pretty good job of talking about why is the rationale for this project to begin with. But I also just want to throw out here some data that GSI has collected. So these 2 species, Mercenary and Mercenary campechiensis, although they look very similar, and to our eye, a clam's a clam, they are physiologically different, right? And so what you're seeing here is some data that we collected from a ARC project looking at the temperature and salinity tolerance of these 2 species side by side. And what you'll see here is that the boxes are really on the left. You've got kind of their survivorship by temperature at 2 different salinities. And then on the right, you've got a varying salinity of these two species, but keeping temperature at 28 and 35. And in both situations, 2 separate experiments, the through line is that the southern hard clam does better at lower salinity than the northern. Even though the northern actually has a little bit better temperature tolerance at extreme temperatures, the southern hard clam is a little bit more resilient at lower salinities, of which leases commonly do from time to time experience. The point there is, not only are we interested in the southern hard clam because there is tons of money in the state right now for doing shellfish restoration, but what are the things that we're missing because we can't experiment with the southern hard clam because we can't identify a southern hard clam well for the commercial application. So this is always something that GSI has been interested in, is what is the applicability for the southern hard clam for commercial aquaculture. But part of the foundation is making sure that we can tell what species we're working with. So that's really the project basis. So our objectives at GSI are pretty simple. I said we're the fins in the water. We're primarily interested in collecting the brood stock necessary that's going to go into kind of be the reference population for Jan and Kim. Our second thing is to maintain the brood stock. And we've unfortunately learned that that can be harder than we think. We collected a whole bunch of individuals before the storm, Milton and Helene, and unfortunately, all of those were lost following the hurricane. So we're definitely, this step is not lost upon us just because we collect hard clams. If we can't genetically verify them and pass them off to the industry alive as future brood stock, what's the point? And lastly is to certify the brood stock. And I'll get into a little bit about that. Jan and Kim have already kind of talked about the science behind that. So for collection of broodstock, we're going to try to cover as broad of a range of their purported native distribution as possible. So we're going to be making trips all the way up from Pensacola, all the way down to maybe as far south as the Florida Keys. And our process here is to first assess southern hard clam habitat suitability. And I will get to the importance of our local and industry partners that will play in this, because that's a lot of ground to cover. But then it's our job to hop in the water and conduct underwater searches at candidate sites that we think hard clams will be present. So what we've done today and kind of worked through our kinks is we go to a candidate site. We first hop in the water, look around for hard clam shells, which is a pretty good indicator that, you know, possibly live individuals will be there. Lay down a transect, and then two divers will lurk along that 30-meter transect using hand raking and digging to hopefully find some individuals. So once we have live individuals, we'll then transport those back to a holding facility where they will then enter in that kind of maintaining the broodstock process. Okay, so as I mentioned, there is a lot of water to cover. And so part of what I want to communicate today to the industry is that we need your help, and we want you to be a part of the process. So be a hard clam helper. First, I'm going to have two slides. The first is going to be specifically for industry participants. The second one is for just kind of general academic and nonprofit. But you know, if you're an industry member, if you're an active shellfish farmer and grower, we're interested in helping or having you help us facilitate our collection trips. Okay, so we're seeking local watermen and women to provide local knowledge to their area across the West Coast, especially in the areas that we're not as familiar with. And we also would like to help in doing some pre-collection scouting. So, you know, having those local watermen and women go out and maybe not hitting a lot of sites in a local area and providing us intel back as to, okay, here are the best places to go when you do make your collection trip, GSI, to, you know, find some hard clams. And then, you know, we're also, you know, they're watermen and women. You're on the water all the time, right? So the chances that you might come across a hard clam are a lot better. And so we would love your help with that as well. You know, we really value the industry's time. And so, you know, we baked into this you know, we're going to pay you, you know, like you're going out in the water, helping us out. We want to pay you for your services. And so we're looking at $1,500 per watermen and women for those kind of selection services. So it's not all the sites, right? We don't, you know, we feel pretty comfortable hitting some of the local Tampa Bay, Sarasota Bay, we know pretty well. But if you are an industry member who is interested in assisting, please contact Sarah Hutchins. I'll put her information in the chat again, but her email is listed there. All right. And you don't have to be an industry member to help. We're also seeking volunteers to just essentially expand our collection scope. GSI is helping right now with actually FWRI collections. So when we're out looking for hard clams, we're helping FWRI collections look for a trina species because they need a trina species for their collections. So we hope that we can leverage the kind of the marine community here in Southwest Florida to all help each other out. And in fact, actually, one of the good signs of hard clam habitat is finding live atrinas, as we think we're on to. So if you're a volunteer, what do you need to do to collect hard clams? You actually don't need that much. You need a hand rake, a collection permit, and that can be as simple as a saltwater fishing license here in Florida. GPS notebook, and a cooler with ice and cardboard to transport the hard clams back until we can pick them up. I also will say a dive flag. If you're safety first, always. They always seem to be in spots close to boat channels. That's my experience. So make sure you're safe out there. But how to find hard clams. So we're not, this is I'm sure the local watermen and women can give us some pointers, but in our experience, the first thing if you're going to plan a trip out is to identify suitable habitat locations before you go out. So our experience has been locations on the interior of passes, sandy locations, high flow locations near seagrass beds, other bivalve macrofauna, such as, pen shells is a good indicator that you might have some hard clams. And then our process is we hop in the water first with a snorkel, we swim around, and we see if we can confirm the presence of just even dead Mercenaria shells. If you can find articulated shells, that's even better. And then, if we feel like we have a good spot, we'll lay down that transect and then we'll combine hand raking using that hand rake to kind of I find something hard underneath the surface and then use our hands to dig into the sediments. And then, lucky is always helpful. So they call it fishing and not catching for a reason. All right. If you find a hard clam, what should you do? So please note the GPS coordinates of the collection sites. And if you've collected multiple hard clams from different collection sites, try to keep them separate in the cooler if possible. And you want to lay those hard clams down over top, a little bit of ice or freezer packs on top of cardboard. So you don't want to put the clam directly within the ice. You just want to keep the cooler chilled until they can get off the water. And then give us a call to coordinate pickup. And we'll also give you instructions if we can't get into, pickup within that 24-hour period on how to store them. All right, so we've got about 2 minutes left. So what we're going to be doing is then maintaining those brood stock at on-land facilities until we can genetically sample them and then send them off to VIMS. Once we do that, we will then take the brood stock and transfer them to a commercial aquaculture lease, and we'll engrave all of the hard clams on externally on their shell so we know which site they came from and identifying each individual as well. And in the event of any sort of severe environmental perturbation, like a hurricane, this time we are going to extract our precious hard clams from a commercial lease ahead of time and move them to very, very safe situations on land. And I also just want to give a shout out to Dr. Nicole Roddy at Moat Aquaculture Park. She's already provided us space there at her facility to house them, and we really appreciate that. All right, finally, so GSI is going to serve, what we want to be able to do is be a permanent presence here in the state of Florida. to kind of have the ability of somebody in the industry needs to know, hey, I want to collect additional brood stock. How do I do that? Well, we know the places to go to collect brood stock. You have collect some brood stock. Is this a campy? Send us a tissue sample and we will tell you if it is or not, right? So we're going to use that VIMS marker panel to certify campechiensis brood stock. We're also going to provide all the brood stock that we collect and test to hatcheries at no cost and without royalty. So we're going to take the specimens at the end of the project and turn them over to interested hatcheries. And then finally, we're going to make sure that we're always a resource for the industry to answer this very basic question: what species of hard clam do I have? So our vision is that should be able to send us a sample, and we can give you within a reasonable turnaround time and very low cost the answer of whether that is a northern or southern hard clam or hybrid. All right. That's all I got. Turn it back over to Angela.
All right, wonderful. Thank you, Stephen, for that overview. And I'm sure that there are questions. If you have a question, you can either unmute yourself or turn your camera on, or you can use the little hand raise icon if you want to get crazy. That's fine too. Or put your question in the chat. Any of those will work with this small group. I did want to say real quick for the record, and I know that you all know this, but when Stephen was mentioning collecting hard clams in coastal habitats, it's important to remember, obviously, to avoid, remember best practices. So disruption of seagrass habitat is not encouraged, even though hard clams might be found in seagrass habitat. Make sure that you're not raking and disrupting sensitive environmental areas within our estuaries.
Yes. May I also add that I second that. And in fact, I said near seagrass beds. All right. And actually, you really will, even if you were to try to do that, and you should not, because you do not have the correct permit, you won't be very successful because seagrass provides protection. So yes, please do not destroy any existing natural resources to find hard clams.
So we really wanted to use this time to get feedback from stakeholders on how this project looks. If you have opinions on how it should proceed forward, or any suggestions on things that you'd like to see come out of it that you might not have heard. If you have questions about the methods that were presented or how to get involved more directly, please go ahead and use this time.
Stephen, you mentioned that you're pretty comfortable with doing most of the collections within the Tampa Bay area, but would you be interested in volunteers through the estuary program? And okay.
Absolutely. I mean, our intention is to provide like a minimum collection, right? And to the extent we can engage, even down to, just kind of a citizen science thing, right? And I think this is a great candidate for one. Find a hard clam. It's going to be used as a genetic reference. And, Kim, Jan, correct me if I'm wrong, but, we don't know what a true, I mean, there's already been intergression in between these species, right? That like the cat's out of the bag.
Yeah.
So the power here will be collecting as many individuals as possible to provide that reference. Yeah.
Correct. And then we will be able to have a better handle on. Yes.
Okay, I'll connect with you later.
Power will be better the more individuals we're able to get. That's correct. Kurt, you had a question?
Kurt. Oh, you're muted, Kurt.
There you go. Yeah, just a quick comment. Since we've sent animals around to different farmers, not so much large amounts of seed or anything, but we send a lot of research animals over. We just overnight quite a few animals. So, and then when we've been looking for oddball species in the past, we've reached out to different specimen collectors. A lot of times they're experts. So I guess this is more for Stephen in coordinating and Angela. In Panacea, they have a specimen collection group up there. I've contacted the one that's down in Key West that works for there are a couple divers that work for the aquarium down there, the Key West Aquarium. So you don't have to make that long trip, although I could see other reasons you would want to go. But I think, that could be a resource. And then obviously don't discount maybe putting that message out that people, if they do collect some, they could literally just throw them in with a small gel pack or a frozen water bottle into a small cooler, and FedEx them back to you. And FedExing them would be certainly more viable option than, let's say, you going to send somebody from Tampa up to Pensacola, to pick up 12 clams, right? So a lot of these might be small amounts that people are collecting, and we certainly wouldn't want to discourage even small amounts coming in, right? And the last thing I guess to mention would be, it might make sense to go ahead and try to employ the existing leaseholders. So whether it's oyster farmers in North Florida or clam farmers, whichever throughout the West Coast, maybe getting the FDAX list and just sending something out, stating that it's as simple as send us overnight a clam to us, use our FedEx account number or something like that. And there'd be some money, money available. Obviously, it wouldn't be $1,500 for somebody sending you a dozen clams, but knowing that they'd have all the costs covered and maybe pay for a boat trip or more. Thanks.
On that note, I would say that, Stephen, we might want to create a flyer or something and distribute to the associations. I know we've got Tim and Joey on here from CKAA, and Adrienne has also expressed interest in helping her membership, be aware of the project. So using the associations to help distribute the information is probably a good avenue to take also. Go ahead, Tim.
Yeah, we would love to help out with sending an email out in like the Cedar Key area to all the farmers and getting them involved. And I'll try to reach out to the Big Bend area, to like Homosassa, Crystal River, some buddies that I know down there that do shrimp and different things like that, and try to tap into some other resources and see if we can't spread this along that whole coast there. So.
Angela, I think the DJ had a question in the chat that I think is relevant to what Kurt was talking about. He said, can people freeze clams for samples? And so the answer to that is yes. I think that, it's a live clam is going to be more valuable than a dead clam, but there's still value in just having that individual as a genetic reference, even if we
Right.
can't use that in the future for broodstock.
Right, it would be fine for us, we can use it, but you wouldn't have it for broodstock for future, right? That's what Jan and I were just talking about, we can use it. As long as we know where it's from, it would be really useful. Location is important.
I have a comment and also a big shout out for you all doing this project. I think it's fantastic. And I was a little nervous, and I wanted to ask the question that if we find hybrids, and most of the hybrids are probably from, and I don't want to blame the industry, the shellfish industry is probably much mixing all the clams, and they don't know what they're doing, where their sources are coming from. Regardless of that, I want to applaud is the fact that we can map locations of the hybrids, and if we find some pures, we can also map those locations and then keep those stock breeds separate, or actually as much possible to keep it separate. And I think that is the most valuable thing that we're going to be accomplishing with this project. Thank you so much for having the initiative to do this. We need it. When we're talking restoration, this is the kind of work that is really going to be meaningful later on. Thank you.
Can I throw out something? So yeah, just to clarify, like I don't want anyone to get the impression that I actually think that the shellfish aquaculture industry has done a tremendous job of producing genetically verified uh, southern hard clams to date. So it's not that we can't differentiate between the species. It's just that it's the process to do it is not an easy one. And so Kim and Jan are really working on the ability of developing a rapid genetic test so that, it's just a pain for the industry to currently, okay, I've got this clam, how do I figure out how to do this? And so I think, I don't want to put out there that if you're buying a southern hard clam from a hatchery, those individuals have done more than probably what even they've been required to do regulatorily to verify that that's a campy. So I think, Ernesto, the clams you have out in Sarasota, those are campies, and have already been verified through UF. And Wei Ping has assisted with that. So it's just more of a technique thing and making it easier for the industry to check. That's it.
I'll ask another question. For that, you had mentioned that the hatcheries would be able to utilize the brood stock eventually and it would be available to them. How would they go about connecting with you and securing some of that later on? I know we're a little early in the game still, but eventually would that be a GSI conversation? Would that be a conversation with VIMS? So access to the brood stock, I think, is maybe somebody will ask that eventually.
Yeah, so it's our job at GSI to curate the living brood stock until they're ready to be, until they have been verified as native southern hard clam through Jan and Kim's process. Once that's the case, we can begin to transfer those, and we would prefer, to actual hatchery facilities, right? Because y'all, the industry knows how to take care of hard clams probably a lot better than we do. So yeah, I think I think it's just as soon as we know that they're campies, then let's pass them off and use them. So yeah, contacting us, either myself or Sarah Hutchins, that would be good. And we'll be reaching out to hatcheries as well, and saying, hey, we've got these brood stock available to you. Do you want them? So yeah.
I just wanted that recorded so we could listen to it later. I knew the answer, but does anyone else have any other questions?
Stephen, as far as collecting clams and when we're engaging with potential volunteers, are you going to put together some sort of guide as far as appropriate habitat to collect from, a couple of guidelines based on the biology?
Yes. So we're gonna hire an additional staff member that will be kind of along with Sarah primary on this project. But within the next month or two, we want to get that document together, right? So that we can disseminate that to any volunteers. So absolutely, I'm envisioning kind of the slide I have had of, oh, you want to be a volunteer? Here's what you need. Here's how to find them. Here's what to do if you find one. And yeah, we'll share our tips as to how to find hard clams.
What's your timeline on the guide and your collection window?
So our collection window will, expand. We got a little bit of delayed start because of the hurricanes. We're actively collecting right now. We'll be collecting through I mean, that's a question Kim and Jan we need to have, but I certainly through the end of the year, probably into the beginning of next year.
Yes.
We don't, there doesn't need to be a strict cutoff for that for us. I mean, we can keep sending samples for a long time, and then even after that, we'll have to test the marker panel. So there's no, it'd be good to have, a certain subset of samples as early as possible. Beyond that, I mean, I don't think that we have to put a hand on it.
We can continue to collect and get more samples. And
Get the verification process up and running and make sure that's smooth. So yeah, no M for you.
No.
I'm teasing.
keep them going.
And for the industry partners that would like to receive compensation for participation, Stephen, will they like invoice you? Should they work directly with GSI to organize that? Working with universities, there's like a vendor registration process. With GSI, it's a lot more simple. So can you just kind of walk through the process of what that will look like for industry?
Yep. So, if you think the big thing is we want to find guides within areas. So we want to find individuals or watermen and women spread out across the west coast of Florida who are in locations that we're not familiar with. And so if you think that, if someone thinks that they would be a good candidate to provide that local knowledge and guiding, first step is to reach out to Sarah Hutchins. You drop her contact information in the chat. It's also in the slide that I provided. And we'll go from there. Yeah, so it doesn't need to be an involved process. Nice thing about working with small nonprofit is we don't have a lot of the hoops to jump through. And so it could be as simple as agreed upon, here are the services and an invoice when the services are completed.
Go ahead, Kurt.
Oh, just one other comment. I was just thinking, when you set up whatever you do, a flyer or whatever your information you're putting out for collection, you may also want to have a size limit on there, because obviously somebody could collect and or send animals that are, southerns that are 5-6 inches across, and there's none of the hatcheries can use those for breeding purposes in general, since they don't strip spawn well, and they tend not to work well to naturally spawn, or condition, etc. So my suggestion from a hatchery perspective would be limit that size to maybe 3 inches across maximum, or call it a top neck size, which the industry folks would be familiar with. But
Hey,
any
Kurt.
size smaller would be fine.
Oh, I guess that's a good point, Kurt, but I'd like to ask him and Jan if those larger ones would be valuable from a genetics perspective.
Genetics, yeah,
They're
okay,
fine.
for that.
They're fine. Fine for us.
Well, then I guess my only point is you just want to make sure that you aren't just getting large animals, because, and I say that in a very serious manner, because if we go out to look for southern hard clams, we will find many large ones, very few that are of general good breeding size or smaller. So at least somehow inform folks that you're looking for samples of this size, but I guess willing to take larger samples. I just hate to see, I mean, it's great to map out the entire West Coast, but the goal of distributing broodstock would be a whole nother situation, right? Thanks.
Okay, we have about 3 minutes until the end. So I wanted to see if Kim and Jan or Stephen and Kurt have any final remarks or closing comments that they'd like to share.
I just want to thank everybody for Enduring molecular jargon. Thank you.
Yeah, and thank you for the support.
Yes, thank you.
Yeah, and also a pre-thank you to the industry. You know, I think the success of this project will largely depend upon y'all's participation, and I think it'll be exciting to have a large group of kind of a community science project along the West Coast.
All right, fantastic. Well, we will be sharing again a recording of this presentation as well as copies of the presentations with anyone, and it'll be available on our website probably within the next week or so. I'll send a link to everybody who's on this call now, but also we'll be able to share it within our networks, and feel free to forward the link to the materials to whoever you think should see it. And in the meantime, all of the information and links to all of these folks' contact info is in the chat. So feel free to copy and paste that if you like, and we'll be in touch for sure. Thank you guys for coming.
Thank you.
Gonna pull up our calendars. No, 15, 16, 17.
Okay, I think that went well. How do you guys feel?
I think it went good. I think it was good. It went well.
You did an excellent job.
Hang
Thanks,
on,
you
hang
did,
on.
you did.
Hang on, don't say anything. Let me stop recording so I don't have to figure this out later. Okay, there.
Well, now